Yeah. So today we are going to deal with the chapter cell cycle and cell division. So to learn this chapter cell cycle and cell division clearly, you have to be clear with cell, the unit of life. We have learnt about all the organelles. Let me put few questions to know your understanding and test your memory. That we have learnt about prokaryotic cell, right? First let us start with the basic questions. Okay. Tell me what is the smallest living organism? Small. Does it have wall or not? Cell wall? No. Okay. Is it pleomorphic? Yes. Yes. What is addressed as jokers of plant kingdom? Largest cell? Longest cell? Who discovered living cell? Who gave us modified cell theory? Rudolf Virchow. In the year? 1855. Good. You are clear with cell theory, cell. Tell me what are the exceptions of cell theory? Good. And then give me an idea on how many types glycocalyx is composed of? One is? Second is slime layer. Cell wall of bacteria is composed of? Cell wall of plant cell is composed of? Cellulose. Only cellulose? Hemicellulose, pectin. Cell wall of algae is composed of? Mannans, galactans, silanes. Okay, with these the cell wall of algae is composed of. Now give me an idea on the composition of fluid mosaic model of plasma membrane. It is composed of proteins. Who is that who is saying vitamins? No. Proteins, lipids and carbohydrates. Carbohydrates are in the form of glycoproteins and glycolipids. Tell me what is the percentage of proteins and lipids in the plasma membrane or cell membrane? Proteins and of lipids. Good. Tell me what is a major function of plasma membrane in transport, types of transport? Passive. Okay. Which mode of transport requires energy? Pinocytosis is also known as pinocytosis. Cell? Cell drinking. Phagocytosis? Okay. You're clear with transport, clear with cell wall. Tell me, if I have to take a plant cell from plasma membrane to outermost, which layer comes out? Tell me that hierarchy. Plasma membrane, close to plasma membrane, what layer of cell wall will be? Very good. Who is that? Very good. Secondary cell wall followed by primary cell wall. This will be the order from the plasma membrane to the outermost layer. After primary cell wall, it will be middle lamella. Amongst these three cell wall layers, tell me which cell wall layer is thin, extensible and it disintegrates with the increase of the cell size? Primary cell wall. Tell me which cell wall layer will have pectin and suberin depositions? Secondary cell wall. Okay. And now what is the common non-membrane bound organelle observed both in prokaryotic cell and eukaryotic cell? Ribosomes. Clear, right? It is ribosomes. In eukaryotes, what type of ribosomes do you find? What is the role of ribosomes present in any cell? Okay. Tell me about single membrane bound organelles. Endoplasmic reticulum, Golgi bodies, lysosomes and vacuoles, which form endomembrane system. Their functions are coordinated. Amongst these four organelles, tell me which organelle is involved in packaging and sorting of proteins? Golgi bodies. Which organelle is involved in synthesis of proteins and lipids? Endoplasmic reticulum. Which organelle is involved in digestion of molecules? Lysosomes. Which organelle acts as the dump yard of the cell in a plant cell? Vacuoles. It occupies how much percent of volume? Okay, it is surrounded by what layer? How molecules are transported into vacuole? Via active transport. Fine, right? You are clear with endomembrane system. One small question about Golgi again. Okay, tell me which organelles will have which type of cells will have higher amount of Golgi bodies? Which types of cells? I told you few cells will have abundant Golgi bodies, few cells will not have Golgi. Acrosome of sperm cell, root cap cells to enhance the positive geotrophic movement. And then about double membrane bound organelles we have learnt. What are the types of plastids? Chloroplast, leucoplast. Tell me which type of leucoplast will store the protein substances? Protein, alluroplast. Carbohydrates will be stored in amyloplast. Okay. Now we have also discussed about sites of reactions inside the chloroplast. Tell me where does light reactions occur exactly in the chloroplast? Thylakoid lamellae. In grana, exactly in thylakoid lamellae. Glucose is produced in stroma of the chloroplast. Clear with the chloroplast? Mitochondria. Simple question about mitochondria. It is also known as powerhouse of the cell. Where is ATP synthesized in mitochondria? Matrix of mitochondria. Second question. All the electron carriers, protein channels, whichever are required for the synthesis of ATP, where are they present? Inner mitochondrial membrane or cristae. Fine, right, beta? So this is about mitochondria. Tell me about those organelles which has their own DNA. Mitochondria and chloroplast. They are addressed as self-replicating organelles or semi-autonomous organelles. How do they divide? By binary fission. By binary fission. Okay, fine. We'll pause here for a while. We have discussed that these two organelles are semi-autonomous organelles, but they share few traits with prokaryotic cells, be it in the type of division, that is binary fission, be it in having double-stranded circular DNA, okay, or be it having 70S ribosomes. These are the traits that it is sharing with the prokaryotic cell. Why? Why? Can anybody answer? One time asked question, previous in your PYQs, if you see, there will be a question on this. It is because of? No, it is explained in a theory called as endosymbiotic theory. What happened is, if you look into the evolution, okay, Miller experiment and all, where you will find how the molecules have existed in the beginning. So what are the ancestors of the complete life, complete living organisms? First ancestors were prokaryotes. We address them as archaebacteria. Okay, so along with this bacteria, these are also the prokaryotic cell in very past. Okay, so in due course of evolution, when eukaryotic cells have enhanced, these eukaryotic cells have engulfed these mitochondria and chloroplast cells. Okay, and then mitochondria and chloroplast have become part and parcel of eukaryotic cells. So previously they were prokaryotic cells, though they have become part and parcel of eukaryotic cells, they share some traits. This is being explained under the concept of endosymbiosis under endosymbiotic theory. So they share few traits of having double-stranded circular DNA, 70S ribosomes, and dividing through the pattern of binary fission, and that is how they exist as independent cells as well. Both the organelles chloroplast and mitochondria will synthesize ATP. Okay, if you look into the Kelvin cycle, C4 cycle and all, though chloroplast is not addressed as powerhouse of the cell, it is known as kitchen of the cell because it is providing food, but then ATP is synthesized here as well. Energy is synthesized here as well, even in mitochondria. Fine, right? Understood about this? Yeah. And then about nucleus. Whenever you are going through nucleus, you should be clear on the chromosome structure, different shapes of chromosome. V-shaped chromosome is also addressed as metacentric chromosome. In which phase of cell cycle you will see clear structure of the chromosome? Metaphase. Shapes of the chromosome? Anaphase. And then tell me, in which type of chromosomes centromere is present close to one end? Close to one end. Acrocentric. very close to telocentric, where one arm of the chromatid is completely negligible. This is about shapes of the chromosomes. Tell me, you have also learnt about cytoskeletons, 3 types of proteinaceous filaments: microtubules, microfilaments, intermediate. Amongst these 3 types, tell me which proteinaceous network or filaments are keenly involved in cytoplasmic streaming? Microfilaments. Which are involved as a constituents of spindle fibers? Which are involved in mitosis divisions? Microtubules. Okay, which are involved in cell division? Microtubules. Cell plate formation? Microtubules. Remember that. That is about cytoskeleton. And then we have also learnt about cilia and flagella. Prokaryotic flagella will have what microtubular arrangement? 9 plus 0. 9 plus 0 microtubular arrangement. And what is a protein that is present in prokaryotic flagella? Prokaryotic flagella. Flagellin. Eukaryotic flagella will have tubulin. Okay, microtubular arrangement of eukaryotic flagella 9 plus 2. Total number of microtubules present in a centriole? 27 microtubules, because there are total 9 radial spokes. At the center there is no microtubule, you just have a hub to which 9 radial spokes are present. Each radial spoke will have 3 microtubules. So on total there are 27 microtubules in a centriole. Tell me how many number of microtubules are present in a cilia and flagella? 20. Okay, at the center you have 2 central microtubules which are enclosed in a central sheet, to which 9 radial spokes are present. Okay, each radial spoke will be having 2 microtubules. So on total it is 18 plus 2, 20 microtubules. Clear? Once reframed everything, recalled everything. Now about cell cycle and cell division. This is a very small chapter. At least one question for sure it will come from this chapter, be it in NEET or EAPCET. One question for sure. Okay, what you have to learn from this chapter? Firstly, tell me what is cell cycle. What is cell cycle? What do you learn in cell cycle? In cell cycle you will learn all the sequential events, complete events. You will learn it in sequence, starting from duplication of a genome. The genome which is present in each cell will be duplicated. Synthesis of new cellular constituents or cellular organelles, followed by duplication of 2 daughter cells or formation of 2 daughter cells. All these events you will learn it in a sequence. So this complete events put together, they form what? Cell cycle. What do you learn here, ma? You learn about, these are the sequential events starting from genome duplication, synthesis of new cellular constituents and formation of daughter cells. Fine, right? Clear, Noma? So from the duplication of genome to synthesis or formation of daughter cells, all events comes under cell cycle. Whereas cell division includes only that phase where a cell divides into 2 halves. So this includes only M phase. Okay, all these events are covered in 2 phases. Cell cycle is covered in 2 phases, which is interphase, which is called as I phase, and M phase. Whereas cell division includes only M phase. You are clear till here, no? What are the events that occur in cell cycle? It occurs in 2 phases, interphase and M phase. Cell division, only duplication of cell. Division of cell occurs in only M phase. Once this is done, you see next. So here if you see, interphase includes 3 subphases again, which is G1 phase, S phase and G2 phase. This is under interphase. M phase includes karyokinesis and cytokinesis. And cytokinesis. So here if you take down this one chart now, you will understand everything. Yeah. Cell cycle is included under two phases, which is known as interphase and M phase. Okay. So interphase includes three subphases inside it, which are G1, S, G2. M phase, depending on the cells, whether it is somatic cell or germ cell it is happening. We have two types of M phases. One is mitotic phase, meiotic phase. Mitosis, meiosis. Fine, right? So mitosis, again karyokinesis happen. Karyokinesis means movement of nucleus. Cytokinesis will happen, that is cellular division. Karyokinesis is divided to 4 sub-stages: prophase, metaphase, anaphase, telophase. Cytokinesis is different both for plant cell and animal cell. Done, right, ma? Then you observe meiosis. Meiosis occurs in 2 stages. We call it as Meiosis I, Meiosis II. In Meiosis I also what takes place? Karyokinesis, cytokinesis. Karyokinesis 1, cytokinesis 1. Okay. Karyokinesis 1 and cytokinesis 1 are interrupted by a short stage in between them, which is addressed as interkinesis. Okay. Next is Meiosis II. It again has what, beta? Karyokinesis 2, cytokinesis 2. Fine, right? So karyokinesis 1, you have what? Prophase 1, metaphase 1, anaphase 1, telophase 1. Fine. So at the end of cytokinesis 1, what happens is 2 daughter cells are formed. Which are interrupted by what? Interkinesis. This is the shortest phase between Meiosis I and Meiosis II. Prophase 1 is the longest phase, wherein here you have 5 substages under prophase 1. If you take 10 previous year questions of NEET, 3 to 4 previous year questions will be just on the prophase 1. Okay, wherein it has 5 substages, which includes leptotene, zygotene, pachytene, diplotene, dikinesis. Karyokinesis 2 includes prophase 2, metaphase 2, anaphase 2, telophase 2, followed by cytokinesis 2. At the end of cytokinesis 2, here each daughter cell will again divide to 2 cells. At the end, how many cells are formed from a single parent cell? 4 daughter cells are formed. So now you got a clear picture on what we are learning in this chapter. Okay, very small, no? This has fit in your mind. Drop your pens, everybody. I'll give you 2 minutes time to copy, no problem. Drop your pens, everybody. So in cell cycle, we are learning everything in the form of sequential events, beginning from duplication of genome, synthesis of cellular organelles, followed by division of daughter cells or formation of daughter cells. Includes under, this is done under 2 phases, interphase and M phase. Like how you are preparing for 2 long years, taking 1 more long term to 3 long years to attempt for 1 exam, that is 3 hours. Same the case, the complete preparation for division happens in interphase. And what are the molecules that are, what all are the molecules that are required for the synthesis of the division will be synthesized here. That is the reason interphase is also addressed as preparatory phase or biosynthetic phase, and this occupies 95% of cell cycle time. Also addressed as what ma? Preparatory phase or biosynthetic phase. And it is divided into 3 substages, that is G1, S and G2. Now M phase, it can happen in 2 different phases, mitosis and meiosis. Mitosis is also known as equational division. This is operated in all the somatic cells of the living organism, okay, wherein chromosome number remains same. For example, the parent cell is having 24 chromosomes. Daughter cell also will have 24 chromosomes. As there is no reduction in the chromosome number, this is also addressed as what ma? Equational division. Okay, occurs in what type of cells? Okay, it occurs in only diploid cells of animal cells, occurs both in the haploid and diploid cells of plant cells. Okay, in plants, both in the haploid and diploid cells, mitosis will happen. In animals, only diploid cells will undergo mitosis. Now let me ask you a simple question. What is haploid, what is diploid, what is ploidy? Can anybody tell me? Don't say me this, ma'am, this is addressed as haploid, this is known as diploid. Don't say me that, please. If that is the picture in your mind, don't say me. If any other picture is there, you can tell me. No, there is no relation with nucleus, number of nucleus, because if single nucleus, that is mononucleate, 2 nucleate, binucleate, many nucleate, polynucleate, since ITM condition, there is no relation with that. Ploidy has a relationship with number of chromosomes. In a single nucleus, see beta, let's suppose a single cell, your cell only. How many chromosomes are there inside a single nucleus, inside a single cell? How many set of chromosomes are there? 23 pairs. Total how many? 46 chromosomes. Out of 23 pairs, only 1 pair is sex chromosomes or germ cells. Okay, only 1 pair is of allosome. Next all are what? Autosomes. So in a single nucleus, 23 pairs are there. The topic of ploidy lies with chromosome number. There is no relation with nucleus. Tell me. Very good. What is your name? Good. Gangotri? Yeah. So see here. Haploid means if a cell, any cell is having half set of chromosomes. For example, 46 is the set of chromosomes. If you have only 23 set of chromosomes in a cell, which means you have half the set of chromosomes. It is known as what? Haploid. Diploid means complete set of chromosomes. Complete set of chromosomes. Be clear with this, ma. Nobody will tell you when you go to higher standards. Don't give blank faces when you go to graduation when they ask you on ploidy. Okay? Haploid means this, diploid means having complete set of chromosomes. Clear with this? So mitosis occurs in diploid cells of animal cells. In plant cells it occurs both in the haploid and diploid cells. Clear with mitosis? First nuclear division, nuclear movement will happen. Karyo means nucleus. Kinesis means movement or division. Okay? So nuclear division is addressed in 4 stages: prophase, metaphase, anaphase, telophase. Followed by cellular division, that is cytokinesis. Occurs differently in plant cell and animal cell. Already plant cell cytokinesis we have discussed. Okay. Next about meiosis, which occurs only in the germ cells, and meiosis is known as reductional division. Why? Because chromosome number gets half in the daughter cells. For example, if a parent cell is having 46 chromosome, daughter cell will have only 23 chromosomes. Chromosome number is reduced to half. That is known as meiosis. It happens in two phases, meiosis I and meiosis II. Meiosis I, karyokinesis 1, cytokinesis 1. Karyokinesis 1 takes place in 4 substages, 4 stages: prophase 1, metaphase 1, anaphase 1, telophase 1. Prophase 1 has 5 substages again: leptotene, zygotene, pachytene, diplotene and diakinesis. You can remember in this order of LZP D square. Okay, LZP D square. Leptotene, zygotene, pachytene, diplotene, diakinesis. Next, cytokinesis 1. 2 daughter cells are formed after this. Interkinesis followed by meiosis II. Meiosis II will be same that of mitosis. Same as that of mitosis. You got a clear picture on this? Okay. And M phase, how much time it takes place? Less than 5% of total time of cell cycle is in M phase. Okay. And cell cycle time varies with different cells. For example, yeast cell completes its cell cycle in 90 minutes. Complete cell cycle means starting from G phase to cytokinesis. It gets completed in how much? In yeast cells, it completes in 90 minutes. In a human cell, 24 hours. Okay. A single cell to divide only, it is taking 24 hours. Okay. Yesterday few people are asking me, ma'am, complete NEET preparation should complete in 30 days. How does this happen? The groundwork, the basic work should begin from long tenure. Okay. If you are starting from your basics right away and it should be finished in 30 days, then no intelligent people will ask that question. See, this is all about cell cycle. Got a clear picture? Fine. Just go through this 1 minute. If you are having any doubt, ask me, ma. If this picture is clear to you, then cell cycle and cell division is in your fingertips. Yes, Sushmita, meiosis is reductional division. Done, right? Yeah. Now we will move to the types of cell division. So far we have learnt about 3 types of cell division. One is what? Mitosis and meiosis. But there is one more type of cell division which is the first and foremost basic type, which is known as amitosis, which is also known as binary fission. Means division without the help of any spindle apparatus formation. Karyokinesis and cytokinesis will happen parallelly. Okay, there is no completion of karyokinesis and then cytokinesis will start. There is no sequential phases. It divides as it is. 2 cells will divide like this. Okay, this is binary fission, happens in unicellular organisms. You can highlight the examples like unicellular organisms and mitochondria and chloroplast also divide by binary fission. Okay. No spindle apparatus or spindle fiber formation. And then we have discussed about mitosis and meiosis. Clear, right? Then about interphase. Remember, G1 phase is also addressed as, if you can see here, G1 phase, can I name G1 phase, pre-synthesis phase and post-mitotic phase? Can I name it so? It is pre-synthetic phase, post-mitotic phase. Same the way, how can you name S phase? Pre-G2 phase, post G1 phase. Same the way how you will name G2 phase, pre-synthetic phase, post mitotic phase. No, it is pre-M phase and post-S phase. Okay. How you can name M phase? It is pre-G1 phase, post-G2 phase. Clear, right? Yes or no? Take the synonyms properly. G1 phase also known as or post synthetic phase is known as pre or post S phase. G2 phase is pre-mitotic phase or post-S phase. Now you see in G1 phase, okay, that is gap 1 phase. After G1, S and G2, I will also deal with checkpoints, okay. In G1 phase, what happens is, most of the cell organelle duplication and synthesis will happen in G1 phase itself, okay. When all the cell organelles, listen carefully, a single cell, it has entered the cell cycle phase. First is G1 phase. In G1 phase only, most of the cell organelle duplication happens. All the cellular constituents whichever are required for the division will be synthesized. So when all these are duplicated, what happens initially? Cell cycle will increase. Yes or no? Cell size will increase. With the increase in cell size and duplication of most of the cell organelles, and for division, for genome duplication, if you have to go to molecular basis, you need replication, fork synthesis and all. That and all also will happen in G1 phase, okay. So most of the cellular organelle duplication, cellular constituents will be synthesized, cell growth happens in G1 phase. The length of G1 phase depends on the type of cell. For example, those cells which are dividing fast, they will have short G1 phase, but few cells like nerve cells and heart cells, will they divide very often? If they divide very often, what happens? Tumors will form. Yes, if they divide very often, tumors will be formed. They don't divide very often. So they will have long G1 phase, and from G1 phase, to escape the cell division, to escape the cell cycle, they will enter into G0 phase. This G0 phase is addressed as quiescent stage. Quiescent stage, okay, where the cells are metabolically active. Cells are metabolically active, but there is no cell proliferation. What is cell proliferation? Increase in cell number doesn't happen. There is no cell proliferation, it is in the quiescent stage, and in the cells are metabolically active. And from G0 phase, if these cells want to get back into the cell division, again they will get back to G1 phase itself. So from G1 they will move to quiescent stage. If they want to get back to the cell cycle, they will enter again G1 phase. So what is the major events that happen? The cell size increases, okay, cellular organelles duplicates, synthesis of cellular constituents. Fine. Whatever RNA, proteins that are required for the cell division, most of them will be synthesized here only. Clear with G1 phase, ma? Yes. Clear with those cells which escape from G1 phase? Those cells which do not divide very often? You are clear on this note that G1 phase tenure depends on the type of the cell. Okay. Next is S phase or synthetic phase. To put in simple terms, okay, only two events that happen in the S phase, which is a major phase again. Here if you see, this is a single cell, let us suppose, and this is the nucleus. Okay. Inside a nucleus, if this is the genome that is present, okay, and these are the centrioles that are present in the cytoplasm. This is inside the nucleus and this is cytoplasm. In S phase what happens is, DNA gets doubled, but there is no increase in the number of chromosomes. Of course, you will see the structure of chromosome in prophase, but for your understanding and to tell you about the statement which is given in the NCERT that DNA duplicates but there is no increase in chromosome number. Chromosome structure only you will see in which phase? Prophase. But to make you clear that only DNA increases, I have drawn this. Okay. The number of chromosomes here are 2. If you consider chromatids are 2. Here chromatids are 4, but number of chromosomes are also 2. How you will count number of chromosomes? Based on the number of centromeres. Based on the number of centromeres, you will count the number of chromosomes. 2 chromosomes, 2 chromosomes. DNA gets doubled, but there is no increase in chromosome number. If you see the centrioles, centriole duplication happens in the cytoplasm. In cytoplasm, these are the centrosomes that are present. 2 centrioles get into 4 centrioles. They are perpendicularly arranged like this. Because for each cell division you require one centriole, right? The centrosomes itself will move to the opposite poles in the prophase. So that duplication happens here itself. And with the increase in DNA number, in DNA content, for example, here 2C amount of DNA is there. Here amount of DNA will be 4C. Okay. When DNA is doubling, nucleus is the same, cell is same. In that case what happens? The negatively charged DNA has to be in the precised nucleus only. In that case it has to condense. For that purpose, histone protein synthesis also happen in the S phase. Histone protein synthesis also happen in S phase. Simple events of S phase: DNA duplication, centriole duplication in cytoplasm. For the DNA which is duplicated to stay in the same nucleus, histone protein synthesis happens. 3 events. What are the organelles that are duplicated in cytoplasm? Centrioles and centrosome. What is that is duplicated inside nucleus? DNA. Okay. Duplication, replication, one and the same. Cell division, cell proliferation, one and the same. Next is G2 phase, which is very simple. Listen carefully. In G2 phase, major organelles that are required for the cell division are synthesized or duplicated in G1 phase itself. But semi-autonomous cell organelles or self-duplicating cell organelles, what are they? Chloroplast and mitochondria. They are synthesized or duplicated in which phase? G2 phase. Okay. Semi-autonomous cell organelles like chloroplast, mitochondria are synthesized in G2 phase. Along with this, Golgi bodies are also synthesized. Golgi bodies or Golgi apparatus is one and the same, or Golgi complex. Golgi complex, Golgi bodies, Golgi apparatus, one and the same. And whichever little amount of RNA and proteins are also required will be synthesized in G2 phase. followed by tubulin protein synthesis. Tubulin protein synthesis. Fine. Clear on G1, S and G2? Done? So now get back to this. Tell me in which phase of the interphase most of the cell organelle duplication happens. Tell me in which phase histone protein synthesis happens. Genome duplication? Genome duplication. Genome duplication or DNA duplication is one and the same. Now tell me genome duplication happens in. Got it? No problem. Correct yourself and learn. Next, semi-autonomous organelles are duplicated in G2 phase. Next, centriole duplication. Histone protein synthesis. Tubulin protein synthesis. Golgi bodies duplication. Now, everything is being synthesized. Do you think they are machines and they will work properly all the time? No. Errors happen there also, which has to be checked properly, whether it is happening properly or not. If not, for example, in place of 2 mitochondria, 4 mitochondria are synthesized. Extreme energy is produced, not required for the cell. Extras not required for the cell. If they are produced under, for example, there is an error in genome duplication, it has to be corrected. Okay. So, for the corrections in between the phases of cell cycle, you have checkpoints. All checkpoints you do not require, but basic idea should be there. In between G1 and S phase, major checkpoint is present, which is known as what, ma? G1 S checkpoint, which is major checkpoint. This checkpoint, in this checkpoint, everything is checked with the help of cyclins and CDKs. Okay. The cyclin at G1 S checkpoint is major checkpoint, which is G1 S checkpoint. Cyclins and CDKs which are present over here is G1 cyclins and CDC2 kinase. G1 cyclins and CDC2 kinase. And in between M and G1 phase, again you will have one more checkpoint, which is known as minor checkpoint, which is G1M checkpoint. Guided with what cyclins? G1 cyclins and CDC2 kinase again. Fine. About major checkpoint and minor checkpoint, what are the cyclins and CDKs present? Have that basic idea enough. Clear about the checkpoints, ma? You are clear on every note of interphase, right? G1, S and G2. Shall we move on to the M phase? You are clear on amitosis? Now about mitosis or equational division. Occurs in how many stages? 2. First what happens? And then? Fine, right. So, here if you see karyokinesis, first stage of karyokinesis is what? Prophase. Remember, the complete chromatin which is like this will get untangled to chromatin threads like this. These are what? Chromatin threads. You call it as chromatids, will be formed in prophase, which is for what? Chromosomal condensation. and these chromatid threads which are formed will be adjoined to the cell membrane like this, which is addressed as spireme stage or bucket stage. If I have to consider this as cell, okay, all the threads will be arranged like this and this will be adjoined to the cell membrane. That is why this is addressed as what ma? Spireme stage or bucket stage. Okay, and at the end of prophase, you tell me, if for example complete furniture, okay, all the benches are fit in the room, we can walk freely or not? We cannot walk freely. If all the furniture is removed, we can do walking, we can do jogging, we can do whatever we want. We can roll from this corner to that corner also. Okay, so whatever furniture is there inside the cell will be removed off in the prophase, like disintegration of in prophase, at the end of prophase, we call it as late prophase. Disintegration of nuclear membrane, nucleolus, endoplasmic reticulum, Golgi bodies occur in the prophase last, which is the late prophase. Then the complete space will be there. Now the chromatid threads which are formed can dance from here to there. Okay. So what is that happening in the prophase? Chromatid threads are formed and then chromosomal condensation, spireme stage where chromatids will be adjoined to the cell membrane, followed by disintegration of nuclear membrane, nucleolus, endoplasmic reticulum and Golgi bodies. One more thing, the cytoplasm get condensed here. Why and how? If I have to use, in your IP answer might be you have learned, disappearance of nuclear membrane, nucleolus, endoplasmic reticulum means and Golgi. This statement might be you have read it. Disappearance of all these means they are gone somewhere else or what? No, they have lost their structural stability. They have lost their structural stability, but the constituents of them are present inside the cytoplasm, which makes the cytoplasm dense, condense. Okay, you have learned cytoplasm get dense. That is because they lose their structural stability and that gets condensed inside the cell itself. Understood about disintegration? They don't go to somewhere else, they don't go to space, they will be inside cell only. Clear about prophase? Everyone? Any doubts anywhere? Now, about metaphase, you see clearly. And one more major event that occurs in prophase is the centrosomes whichever are duplicated in the S phase. Two centrosomes are there, let's say. Okay, two centrosomes are there. Okay, these centrosomes will start moving towards the opposite poles in prophase itself. Centrosomes will start moving to the opposite poles. In metaphase, at the beginning of metaphase, which is addressed as early metaphase, complete condensation of chromosome happens. Chromosomes become thickest and shortest. And the centrosomes which have moved to the opposite poles, they will start radiating out the spindle fibers. Okay, these spindle fibers are attached to the kinetochores which are present over the chromosome. Inside chromosome you have centrosome, right? Centromere, sorry. Inside chromosome the primary constriction is centromere. Attached or adjoined to this centromere you have what? Kinetochores, which are disc shaped structures. To this kinetochores what are present? Spindle apparatus will be present. The asters which have moved to the opposite poles in the prophase, they will start radiating out the spindle fibers which connect to the kinetochores. First what happens? Chromosomes thickest and shortest. Then they start radiating out the spindle fibers which make all the chromosomes align at the common plate, central plate, which is known as equatorial plate. Equatorial plate formation is also known as congression. This is also known as what, Nana? Congression. Congression, formation of equatorial plates. radiating out of spindle fibers. Everything happens in the metaphase itself. And if you see, the length of spindle fiber is long when compared to the anaphase. Clear with the metaphase stages? Any doubts anywhere? No, right? Now, anaphase, if you observe. Okay. In anaphase, there is a complex called as APC complex, which is anaphase promoting complex. What this helps is, this helps in centromere splitting. This helps in centromere splitting. For example, see here, centromeres are all in the equatorial plate here, right? Now, centromere splitting will be triggered at the center, where two arms of the chromatids will move towards the opposite poles. They will move towards opposite poles. When they are moving towards the opposite poles, they move in different shapes based on the length of the arms of chromatids. If you have two equal arms of the chromatids, then V shape, metacentric. If you have one long and one short, L shape they will move. If you have very short, very long arm, J shape. If one extremely short and negligible, one very long, I shape. Like that they will be moving towards the opposite poles. Fine, right? This is about what? Anaphase, centromere splitting occurs. Next is telophase. In telophase, whichever chromatids have moved towards the opposite poles, at the end of anaphase, that means at late anaphase itself, we will confirm that these are two different cells. Okay. Now you tell me, if I have to say there are 4 chromosomes here, yes or no? How many chromosomes are present, Nana? 4 chromosomes are present. If I have to consider this as one cell and this as one cell, in each cell how many chromosomes are present? 4. Is there any reduction in chromosome number? No. Chromosome number is same. So it is equational division. In telophase, what happens is, chromosomes which are in the condensed form, they get tangled again, tangled into chromatin thread. They get tangled to chromatin thread again, and again nuclear membrane, nucleolus, endoplasmic reticulum, Golgi bodies reappears. There they will disintegrate, here they will reappear. These are the events of pro, meta, ana and telophase of karyokinesis in mitosis. Go through it properly. If you are having any doubts, just let me know. Some confused expressions you can ask me. Done? So now tell me, chromosomal tangling takes place in which phase? Telophase. Untangling? Prophase. Tell me, where does congregation occur? Equatorial plate formation. APC, anaphase promoting complex, you will see in anaphase. What is its major role? Splitting chromosoma. It will not it will not make chromosome into pieces here, it will just split the centromere, okay. And then tell me what is pyreme stage? Where the chromatids are joined to the cell membrane. Clear? Simple, right? Prophase, metaphase, anaphase and telophase. Fine. A small homework to you. If I dump everything, what you will learn? Tell me what is the longest phase and shortest phase of You know everything here. You are the toppers of IP. Huh? Prophase is the longest phase and which is shortest? I am asking in karyokinesis. Don't be overconfident, write the questions. Longest phase of mitosis and shortest phase of mitosis. In mitosis I am asking. It is an interfamily competition. Okay. I am asking you which is longest and shortest only in mitosis. Okay. Write it and tell me then tomorrow, no problem. Be patient, tell me tomorrow. Next. About cytokinesis. Fine, right. A small correction, ma. Okay, you listen here, I will tell you about that. First about animal cell. Okay, so in animal cell what happens is, from the peripheral ends to the center, okay, it gets constricted like this. So, as it is from the periphery to the center, the mode of cell division is addressed as centripetal. It is known as centripetal. That is how two cells will be dividing. The furrow is from the periphery and it moves to the center. That is how two cells will be dividing after telophase. Okay. Whereas in plant cell, if you consider these two are different cells, see all have moved to one pole in anaphase. Okay, they have moved to the opposite poles. At the center, a cell plate is formed. See, cell plate is formed. Cell plate is made up of calcium pectate, magnesium pectate. At the beginning, it is formed in the form of phragmoplast that will enhance its length and form cell plate. Then that is addressed as middle lamella after maturation. Middle lamella is the first formed layer of the plant cell wall. Okay, so this middle lamella, it is formed from the center to periphery. From center to periphery, known as centrifugal pattern of cell division. I think I have told you centripetal in the previous class. Sorry, please correct. It is centrifugal. The confusion between centripetal and centrifugal is quite common. Remember one thing, if it is from the center, something which is moving from the center to the periphery, we will address it as centrifugal. Something which is from the periphery to the center, it is centripetal. Remember any one thing, the other is obviously noted. Okay. Cytokinesis is clear to you, animal cell and plant cell. Any doubts anywhere? Yes. If the cell division is from the peripheral end to the center, in animal cell what happens is, the limiting boundary is what, ma? Plasma membrane. Plasma membrane is flexible. Yes, right. So what happens? The furrow is formed from the peripheral ends to the center, and that is how two cells are formed. Okay. This is known as it is from periphery to the center, this is known as what? Centripetal mode of cell division. Cell wall is thick or loose? Thick and tough. We cannot divide it from the periphery. So something has to form from the base. Okay. So at the middle what is formed? Middle lamella. This middle lamella will move towards the peripheral ends. Fine, right. That is moving from the center to the periphery, so it is known as centrifugal. Clear about cytokinesis? Okay, the discussion about cytokinesis will not come again. Just know the difference between plant cell and animal cell. It is same in the cytokinesis of meiosis as well.
Ma'am, centrifugal occur in plant cells.
Yes. Done? Done, right, ma? Now we will move on to meiosis. Of course, in meiosis also we have learnt about what? Karyokinesis, cytokinesis, as you said me. Karyokinesis will again happen in karyokinesis 1, cytokinesis 1. Karyokinesis, how many substages? Only pro. Be louder. Has how many substages? 5. How, what are they? Cytokinesis, you will get 2 daughter cells. Each cell will again undergo division, that is meiosis 1, here it is meiosis 2. Okay. Meiosis 2 you have karyokinesis 2, cytokinesis 2. Karyokinesis 2 again prophase 2, metaphase 2, anaphase 2, telophase 2, followed by cytokinesis 2. Meiosis 2 is same that of mitosis. Clear of what you have to learn? Yeah. Let us get started with prophase 1, leptotin. Be here. Yeah. In leptotin, you can see the chromatid thread formation, tangling or detangling of chromatin? Chromatin gets tangled or detangled? Detangled to chromatid threads, which gives out what stage? Spireme stage or bouquet stage. And these chromatids are clearly observed under light microscope. And you can see the centrosomes start moving to opposite poles. Can you see here? Centrosomes with 2 centrioles will started moving to the opposite poles, and chromatid thread formation also happens. Followed by zygotin. Zygotin is a major stage, I am telling you. Drop your pens, listen carefully. Zygotin is a major stage. Okay? Listen carefully. So, chromatid threads are formed here. Okay? You know how the traits are being copied from our parents? How they are copied? Through genes. Genes are transferred. Where are genes present? Sit straight, everyone. Where are genes present? What do you call a gene as? This chromosome is nothing but DNA structure only. It has packed into chromosome, that's it. Okay? So, over the DNA, genes are present. What a single gene is known as? Single gene will have how many alleles? 2 alleles. For example, I have to consider one character or trait. Tell me anything. Always tall and shorter. Height, color, skin color, hair color, anything is known as what? Character or trait. I am considering 2 characters here. One is skin color and the other is hair color. Understand carefully, ma, very important. This understanding is very important. Skin color I am coding, it has 2 alleles. Capital S is the dominant allele, or as skin color is majorly dependent on melanin pigment. More the melanin, more dark color, right? So, capital M and small m. Capital M is dominant, small m is recessive. Hair color, brown we will consider. More amount of B, like it is dominant, gives you black, and small b is recessive. These 2 are the alleles of this character. One character or trait is coded in one gene. One gene has how many alleles? 2 alleles. Okay. Now, for example, here there are chromatids. In these 2 chromatids, here capital B is there, here small b is there. Can they cross over easily? No. So, in zygote what happens is all the chromosomes which have same alleles at same location will come close together, forming out homologous chromosomes. To cross over, they have to come to one place, no? Okay. So, wherever they are different, okay, they are at different places, they will come to one place, forming what? Homologous chromosomes. For example, here you have capital M and small m, capital B, small b. They have come to one place. Fine, right? And these two chromosomes are known as what? Homologous chromosomes. You should be clear on the term of homologous chromosomes and non-homologous. Everywhere you will be used with these terms in genetics, in cell biology. Okay? Homologous and non-homologous should be clear to you. What are homologous chromosomes? If you are not clear with terminology also, no problem, but you should know the meaning. What are the homologous chromosomes? If the alleles, two alleles of a single gene are present at the same loci on two different chromosomes, then those two chromosomes are known as homologous chromosomes. Okay? Two alleles of same gene present on two different chromosomes is addressed as what, ma? Homologous chromosomes. Now you can clearly say what are non-homologous, no? Two chromosomes on which the alleles are at different positions. Okay? These are also known as allelic combinations, and non-homologous chromosomes also known as non-allelic chromosomes. Allelic, non-allelic. Clear, right? So you are clear on homologous chromosomes. In homologous chromosomes, you will come across two different types of chromatids. One is sister chromatids, second is non-sister chromatids. Two chromatids of a single chromosome are known as sister chromatids. In simple terms, you can say two siblings of a single mother. Okay, two chromatids of a same chromosome are known as sister chromatids. Cousins. Okay, non-sister chromatids. Chromatids of two different chromosomes are known as non-sister chromatids. I can take these two, right? So you are clear on what are homologous chromosomes, what are sister chromatids, what are non-sister chromatids, how crossing over happens, what is the story behind. Okay, you are clear with this. Now, in the exam point of view, we will learn. Okay, so in zygote, the formation of bivalents occur. What is bivalent chromosomes? Bivalent chromosomes means it has 4 chromatids. See, 2 homologous chromosomes are coming at a place. Can you see, ma? 2 sister chromatids, homologous chromosomes have come to one place. This homologous chromosomes coming to one place is addressed as formation of bivalent chromosomes or synaptonemal complex formation. 2 homologous chromosomes adjoining together like this is known as what? Synaptonemal complex formation. Bivalent chromosomes. Single bivalent will have 4 chromatids. So, a single bivalent will have how many tetrads? Single tetrad, which has 4 chromatids. So, in your exam, listen carefully. Whenever a term comes on homologous chromosome formation, occurrence of tetrads, formation of bivalent chromosomes, occurrence of synapses or synaptonemal complex formation, anywhere these 5 terms are used, you have to choose Zygotine. Clear about zygotine? Everybody will tell you these 5 terms and they will move to the next substage. But the question mark on homologous chromosome will stay with you for a long term. When I again use the same term on molecular basis of inheritance, it will be a big question mark to you. It should not be like this. So I gave you the complete explanation. Clear? Leptotene, zygotine, you are clear with this? Shall we move on to packetine? Yes? In packetine, now 2 homologous chromosomes are close together, allelic chromosomes are close together, alleles of a single gene are close together. Okay. Now it is easy to cross over or not? Yes. Yes. So in packetine, recombination or crossing over is one and the same. Okay. Recombination or crossing over occurs between sister or non-sister chromatids? Non-sister. Why in between non-sister chromatids, can anybody tell me? Recombination. That's what I am saying. Recombination occurs in between non-sister chromatids. Why? Why? If you can see here clearly, okay, 2 alleles are present on 2 chromosomes. These 2 alleles are of a single gene. Okay, 2 are different alleles. Different alleles, when they combine together only, no, they will give you a new trait. For example, capital M and small m are present. Fine. Then in the daughter chromosome, it will be in the modified version. For example, in the parental chromosome, capital M, small m, small m. Father is of dark color, mother is of fair color. Let us suppose. Okay. Daughter or son, their offspring will be of this allelic combination, will be of brownish color. The fairness will be decreased based on the melanin pigmentation. Single chromosome is extracted, right? Single gamete will have single chromosome. Post fusion only meiotic division starts. Fine. Understood now, everybody? Any doubts anywhere? Yeah. So here in packetine what occurs? Recombination or crossing over in between non-sister chromatids of homologous chromosomes. Between non-sister chromatids of homologous chromosomes. That statement is very clear, and explanation for each statement. Understanding of each statement is very necessary, because most of the questions will be of statement based and assertion reason if you have to take 2025 paper. Previously, if you read line to line NCRTs, you used to get nice marks, but now it is not like that. Understanding of each line is very important, because most of the questions are statement based assertion reasoning. Okay, you should know why, what, how, where is it happening. Okay, so recombination or crossing over is in between non-sister chromatids. And this crossing over or recombination is energy dependent process or enzyme mediated process? Enzyme-mediated process. What is the enzyme that is mediated? Very good. This is enzyme mediated process. By the enzyme recombinase. Fine. And then you can see the recombinant chromatids which are formed. Fine. After which comes the diplotin stage. Now, after the recombination which happens, only leaving the point of recombination, the chromatids will start moving apart from each other, right? Chromatids will start moving apart from each other. When they are moving apart from each other, see, these are the recombination points. And then they are moving apart from each other. When they are moving apart from each other, this forms a chiasmata or X-shaped structures. Okay, X-shaped structures or chiasmata is formed. X-shaped structures are chiasmata formation, where the chromatids will move apart. And in diplotin stage, in this diplotin stage, few oocytes of mammalian cells will stay for long period of time. For example, if there is a female fetus in the mother womb, okay, oocytes which are formed will stop their cell division. Some hundreds of egg cells will be formed when the fetus is in the womb itself. Yes or no? Hundreds of egg cells are formed. When the female reaches at the puberty stage, every month each egg cell will be dropped. Yes or no? Okay. So those oocytes are formed and stopped at which stage of cell cycle? Diploid stage. Sorry, diplotin stage. They are stopped at diplotin stage of cell cycle. X-shaped structures are observed in which stage? Diplotin. Formation of chiasmata occurs in? Diplotin. Then last substage of prophase 1 is diakinesis. Remember the major events that occur in diakinesis. First is terminalization of chiasmata formation, or terminalization of chiasmata, you can say. Okay. Disappearance of nuclear membrane, nucleolus, endoplasmic reticulum, Golgi bodies occurs in diakinesis. Fine. Terminalization of chiasmata means it will lose out that X-shaped structures. Here you ask me, any doubts? Now, as homologous chromosomes are formed, you have two sets of chromosomes with sister and non-sister chromatids. It forms a single metaphasic plate or bimetaphasic plate. It forms a bimetaphasic plate. Can you see here? Bimetaphasic plate is formed. Chromosomes comes to the equatorial plate. The phenomena is known as congregation. All chromatids will underlie at equatorial plate. Okay. To the kinetochore, spindle fibers will adjoin. Okay. Spindle fibers will adjoin, and the centrosomes will move towards the opposite poles in which is substage of prophase 1? Leptotin itself. In leptotin itself, right? So the stage is as it is, but here you can see bimetaphasic plate, that's it. You can see sister and non-sister chromatids of the homologous chromosomes. In anaphase 1, major difference to be noted in anaphase 1 is there is no centromere splitting. There is no centromere splitting means APC protein is active or inactive? Anaphase promoting complex is inactive. If you can see here, sister chromatids of each homologous chromosomes will move towards the opposite poles. Sister chromatids of each homologous chromosomes moves to opposite poles. Next is telophase 1. In telophase 1, what happens? Reappearance of nuclear membrane, nucleolus, Golgi bodies. And if you see the chromosome number, here if I have to suppose this as single cell, you have complete set of chromosomes, yes or no? In anaphase 1, half the set of chromosomes have moved to the opposite poles. So, reduction in chromosome number happens in anaphase 1 only. Telophase, reappearance of nuclear membrane, nucleolus, endoplasmic reticulum, Golgi bodies will happen. Two daughter cells are formed. These daughter cells are haploid or diploid, ma? Haploid. Quick recap. Drop your pens. In leptotin, spireme stage is seen. Untangling of chromatin into chromatids. Under light chromosome, you can visualize these chromatids. Centrosomes will start moving towards the opposite poles, and they start out radiating the spindle fibers. In zygote, formation of homologous chromosomes, bivalent chromosomes are formed, occurrence of tetrad, synaptonymal complex formation, synapsis occurs. Recombination or crossing over by the enzyme recombinase in between two non-sister chromatids of homologous chromosomes occurs in packetine stage. Diplotin, occurrence of X-shaped structures or chiasmata. Okay, in this stage only most of the oocytes of mammalia will be stopping their cell division. Next is diakinesis. Terminalization of chiasmata means X-shaped structures will not be formed. Terminalization of chiasmata and disappearance of nuclear membrane, nucleolus, endoplasmic reticulum and Golgi bodies will. Metaphase 1, occurrence of bimetaphasic plate. In anaphase 1, important point to be noted, okay, centromere splitting doesn't occur, anaphase promoting complex is inactive. Reduction of chromosome number happens in anaphase 1 only. Sister chromatids of each homologous chromosomes will start moving to the opposite poles. Telophase 1, two daughter cells will be formed which are haploid. Clear, right? Any doubt anywhere? Yeah. And then meiosis II. So in between meiosis 1 and 2, I said you there is a phase which is called as interkinesis. Okay, in interkinesis what happens? Cells are metabolically active. Okay, there is no DNA duplication, but whichever RNA and proteins are required for the meiosis II are synthesized here. Okay, these are the events which happen in interkinesis. This is the shortest phase. Clear about interkinesis, right? This is also known as intermeiotic phase. Done, ma? You can copy it later. Listen first, okay? Meiosis II. Same like how it happens in mitosis, it happens in meiosis II. Let me highlight this. Zoom it up. Now you can see it clearly, no ma? Prophase 2, same events. Spireme stage formation. Spireme stage. Untangling into the chromatid threads. Centrosomes will move towards the opposite poles. Metaphase 2, single metaphasic plate. Okay, they will align at the equatorial plate. Congregation occurs. Anaphase 2, APC protein is active or inactive? Active. Centromere splitting happens. Different shapes of the chromosomes, chromatids occur. Okay. Telophase 2, reappearance of nuclear membrane, nucleolus, endoplasmic reticulum, Golgi bodies, and two daughter cells will be formed of same ploidy of the cells. Haploid content, that is half the set of chromosomes are formed in anaphase 1 itself, not here. Okay. So at the end you will get 4 daughter cells which are haploid. Clear, right? Easy, no? Now, major differences between mitosis and meiosis. Mitosis occurrence in somatic cells, meiosis in germ cells. Nuclear division occurs only once. Here Twice. At the end, how many daughter cells are formed? Here, how many are formed, ma? Daughter cells are what ploidy? Here, cells will divide more frequently, right? Only germ cells, so less frequently. Occurs in somatic organs. Here, for the formation of gametes. These are only, there are only one prophase, one meta, anaphase and one telophase. Here, two sets of each. Number of chromosomes are not changed, right? Here they are reduced to half. Crossing over in chromosomes doesn't occur. Here crossing over occurs. In which stage? Crossing over in which stage? Pachytene. In zygote, formation of synaptonemal complex occurs. In pachytene, crossing over occurs. This is also known as equational. This is reductional division. Fine? Clear with everything, no? From the beginning? Recall everything carefully. From the beginning, what we have learned in cell cycle, interphase and M phase, about G1, S, G2. Those cells which are inactive, from G1 they will directly move on to G0. Next, M phase. Amitosis we have learnt, mitosis, meiosis. Mitosis, karyokinesis and cyto. Karyokinesis, pro, meta, ana and telo. All the events do you remember? Next, cytokinesis, in which type of cell it is centripetal, ma? Centrifugal in plant cells. Meiosis, meiosis 1 and meiosis 2. What are the 5 substages of prophase 1? That's all. Okay. Clear with all the phases? Yeah. I'll give you 5 minutes of time. 5 minutes of time. Just go through everything from the NCRT and your class notes, whichever we have discussed now, and then we will go on question solving. You can ask me in between also if you are having any doubts. Done? Shall we move on to questions? Done? Yeah. Look at this first question. How many chromosomes will the cell have at the end of G1, S and M phases? C or B? C. How many B? Listen. Okay. Here you can take prophase, metaphase, anaphase, telophase. Okay. Let us suppose G1. G2 also same, no? Here also entire karyokinesis, cytokinesis also it will be the same number, no? Anywhere do you see chromosome duplication again? So here if at the end of G1 it is 2 chromosomes, at the end of S phase also it is 2 chromosomes. DNA duplication occurs but chromosome number is same. In G2 phase also 2 chromosomes. Here also it is 2. He didn't mention meiosis. If he says meiosis, at the end of meiotic phase you will say it has reduced to half. Okay? Now you tell me what is the answer? B. Understood, ma? There is nothing to confuse. This is where you have to apply your knowledge. You know there is no chromosomal duplication, but when you have to apply it for the question, you are unable to. This requires objective practice, that's it. Okay? Next question you see beta, which is asked in NEET 2025. How many meiotic and mitotic divisions need to occur for the development of mature female gametophyte for the megaspore mother cell in angiosperm? This requires so much of concept from sexual reproduction of flowering plants. Remember this question, there I will tell you, because the explanation only takes 10 to 15 minutes of time. You have to be clear on female sexual reproductive structure, from there the story lands. Okay? A female gametophyte is nothing but embryo sac. For embryo sac formation, how many mitotic and meiotic divisions he is asking. One point to highlight you, that the questions from cell cycle and cell division will not be asked directly, just like that. They will be connected with sexual reproduction of flowering plants. They will be connected with the ploidy of plant kingdom. Like that, based on that you will get the questions. Okay. This you can answer. NEET 2024 question. Chromosomes become gradually visible under the light microscope during leptotin stage. Yes. The beginning of diplotin, which is fourth substage of prophase 1, is recognized by dissolution of synaptonemal complex. Diplotin begins with dissolution of synaptonemal complex and then formation of chiasmata. Diakinesis, dissolution of chiasmata or terminalization of chiasmata. So both statement 1 and statement 2 are correct, but there is no proper explanation. If it is assertion and reasoning, what you have to choose? A and B are correct, A and R are correct, but R is not correct explanation to A. Right? Next. Arrange the given statements in correct sequence of their occurrence during prophase 1. Thin thread-like chromosomes with a beaded appearance occurs in which stage, ma? Appearance of recombination nodules, pachytene. Formation of bivalent tetrads, terminalization of chiasmata, appearance of chiasmata. All intelligent people in my class only, very good. Just in single saying you noticed everything, very good. This is how you will get. There is nothing much difficulty about this. Okay, you should be clear on the sequence of stages. You should be clear on the events that are happening in each stage. Sequence it properly. Easy, no? What is the right answer? A. Good. Next. 2024 asked questions. Spindle fibers attached to kinetochore chromosomes. Kinetochores of chromosomes. Very good, ma. Next. Chromosomes become gradually visible under light microscope during leptotene stage. Beginning of synaptonymal complex. Yeah, repeated question. One thing I will be doing, this complete row is team A, this complete row is team B, this complete row is team C. Whichever team is giving the answer, please raise your hand and then give me the answer. Okay? If without raising hand you are giving, then it is -1. Okay, start. Now some seriousness has come to others also who are not responding. -1 to all teams. Nobody raised the hand. Doubling of number of chromosomes can be achieved by disrupting of mitotic cell division. Disrupting mitotic cell division soon after. Okay, chromosome number gets doubled in which phase? In anaphase. Because chromosomes will be splitted, right? In that phase only doubling of chromosome occurs. Can be achieved by disrupting mitotic division soon after his sign, after metaphase. Okay, next question. Okay, option D, good. The dissolution of synaptonymal complex occurs during diploteny. Not this. Raising hand means it should be this. During which stage of mitosis and meiosis respectively does centromere of each chromosome split? In anaphase of mitosis and anaphase 2 of meiosis. Next question. See, I will go with Jyotika because all of you are raising like this. She said like this. So, Jyotika, huh? Right now? No, I have to give one chance to this team also. Yes? 2. Which one of the following is known as quiescent stage? One more thing to be added. We have to get satisfied with your explanation also from the next question. You have to be ready with the explanation why that answer is right. Yeah, this question? Among eukaryotes, replication of DNA takes place in? Why? How? S phase itself. Is there any chromosomal duplication that is happening? Chromosome number remains? In cytoplasm or nucleus? Where does this occur? Okay, fine. Next question. One minute students. Can you notice the difference between 2023 questions and 2024? In 2024 sum of statement based, in 2025 connecting with other chapters also and asking. That is how the NTA paper have evolved. You also should show some evolution. Ratta marke nahi padna NCERT all the time. You have to know the reasons behind it. Okay? Huh? Which of the following stages of meiosis involves the division of centromere? A. Which team here? A. A. No ma'am, you have to just raise your hand. Straight away. Not like this. Straight away. Yes. The process of appearance in... Wait. The process of appearance of recombination nodule occurs at the... Appearance of recombination nodules occurs in packeting. Your answer is? Hey, who is that misleading the answer here? Next, match the following. No, I will not go with your answer. It should be straight like this. Who is the one who is answering this? Okay. What is the answer? A. M phase? Equational division. Very good. Next, G2 phase? Very good. Next, quiescent stage? G1 phase? Next. To her. One. Whichever hand is straight, I will go with them. One you are saying, huh? See, one point to remember here, ma. Whenever statement based questions are asked, be clear on whether he is asking you to identify correct set of statements or incorrect set of statements. Don't go in hurry like that. Okay? Here he is asking you to identify the correct answer. You have to choose correct set of statements. Statement A, tetrad formation is seen in leptotin? No. During anaphase, the centromere split and chromatids separate. Terminalization takes place in pachytene? Nucleolus, Golgi complex, CR reformed in telophase. Crossing over takes place between sister chromatids of homologous chromosomes. Non-sister chromatids of homologous chromosomes. Between, what are the right answer? B and D. One more point also for your identification. Can you see more set of questions on Prophase 1? Yes. Next. Everything is right in front of you, ma. You have to identify and learn properly. Huh? Good. Option 3. Good. During G0 phase of cell cycle, cells are metabolically active. It is correct only. Cells are metabolically active. Incorrect, sorry. They said inactive, no? Statement 2, the centrosome undergoes duplication during S phase of interphase. Both the statements are correct, ma? First is incorrect, second is correct, no? Okay, okay. Option 3. Next. I'll go with her. Diplotene, good. Hey, what happened to team A and team C? Now team B will not be answering, only A and C. 15th question. Last row should answer. You ask for the explanation, okay? I'll go with her. Recombinase, which is enzyme mediated process. This helps in the formation of recombination nodules. Next. Please properly read. Option 2. Okay. Synapses of homologous chromosomes occurs in prophase 1. Yes. In zygote of prophase 1 only, no? Chromosomes become gradually visible under microscope. No, he is asking you for the sequence, right? For that, what you have to do, dear? First you have to name out, right? Homologous chromosomes occur in zygote. This happens in? This occurs in? Packetine. Crossing over means packetine only, no? Terminalization? Now you tell me. Leptotene, zygotine, packetine, diplotene, diakinesis. It is B. What is the answer? Done. Next question. I'll go with her. 3. Bivalent or tetrad formation in bivalent or tetrad formation feature is observed during synaptonymal complex in zygote, which is option B. Okay? Yeah. Next question. Augustine, you can also answer. Yeah. With respect to metaphase, which of the following statements is incorrect? Okay, she said 2. Complete disintegration of nuclear envelope. Chromosomes lie at equatorial place is true only. Chromosomes are highly condensed. True. Metaphasic chromosomes are made up of 4 sister chromatids held together by a centromere. No, right? Only 2 sister chromatids are at one centromere. See. Complete disintegration of nuclear envelope. At the end of late prophase only we will say they started disintegrating. Early metaphase, completion of disintegration, complete formation of chromosomes will happen. Okay? See, this statement is clearly evident that a single centromere will not hold 4 sister chromatids, only 2 sister chromatids. Next. Good. 1. The appearance of recombination nodules in homologous chromosomes, meiosis, are sites of crossing over occurs. Nodules are formed at the sites of crossing over. Lavanya, try out one question at least. Which of the following never occurs during mitotic divisions? Yes. Pairing doesn't occur, crossing over doesn't occur. Next. Not entertained. What is the answer? How you have? Good. Select the incorrect statement with respect to mitosis. Chromosomes decondense at telophase. Yes, they do decondense at telophase stage. Condensation occurs in prophase. Decondensation in telophase. Tangling in telophase. Detangling in prophase. Splitting of centromere occurs in anaphase. All chromosomes lie at the equatorial plate in metaphase. Spindle fibers attached to centromeres. What happened to you? Getting bored or what? No, you should not. 45 questions you have to solve for single subject in NEET. Okay, just 20-25 set of questions. If you are bored, so how? Sit straight, be active. Next question. DNA replication occurs in? Incorrect statement. It is not in meiosis II, no, it is in interphase. Next question. Leave this, match the following because options are not evident. Go with this question. Next. Which stage of meiotic prophase shows terminalization of chiasmata? Next, this question. Good. Fruit fly has how many? 8 chromosomes. During interphase of mitosis, if the number of chromosomes at G1 is 8, what would be the number of chromosomes after S? 8 only. Okay. I have to tell you about two terms now. The cell whichever is ready for meiosis, parent cell of meiosis is known as myocyte. Daughter cells of meiosis are known as meiospores. Myocyte is diploid, meiospores are haploid. Next question. Last set of questions, okay? You have few more questions, that's it. Centriole duplication occurs in S phase. In cytoplasm or in nucleus? Cytoplasm. Next question. Identify correct statement with regard to G1 phase of interphase. Cell is metabolically active. Good. Next question. Match the following. Match the following type. Zygote in? Packet in? Diploid in? Diakinesis? So total? What is the right answer? Next. Good. And then? Superb. Next. Next question. Superb. Last questions. Last question. This you can answer. How many pairs of homologous chromosomes are there? 6. How many number of chromatids? 12. Number of centromeres? 6. Understood? Got some confidence? Yes. That's it. This is how it will be with every chapter. But how much confident you are at the end of the lecture or that at the end of the practice of the questions, same confidence if you should maintain when you are entering into the NEET exam also. Revision is the key. Revision is the only key. Okay. If you are not revising, everything will be volatile. Got it, ma? Yeah. See you in the next class. Meiospores.